1856, an Augustinian friar began experiments on pea plants in the abbey's greenhouse. His discoveries came to be known as Mendel's laws of heredity, and the man himself, Gregor Johann Mendel, became known as the father of genetics. By using simple probability models, Mendel showed the power of statistics and mathematics in genetics. He crossed pea plants producing only yellow seeds with plants producing only green seeds. The first generation hybrid seeds were all yellow. He then crossed those seeds, and the second generation were 75% yellow and 25% green. Mendel showed that each plant carries a pair of hereditary factors for each character, one factor from each parent. One factor is dominant and one recessive. In this case, the F1 seeds, YG, are phenotypically yellow. Mendel's two factors are a form of a single gene, and we call them alleles of the single gene. A blueprint of genes an individual carries is called a genotype. The express character of an individual is called a phenotype. Common examples are human hair colour, eye colour, and blood types. The familiar ABO blood system is an interesting genetic model in itself because there are three different alleles. Basically, the A, B, and O allele. The A and the B allele are equally dominant, and the O is recessive.
I'm Melanie Barlow. I'm a laboratory head at the Walter and Eliza Hall Institute, which is a biomedical research institute in Australia. I have a PhD in statistics. I work a lot on genetic mapping, finding genes that cause disease in humans, but I've done quite a lot of work in mouse genetics as well, like, for example, genes that determine coat colour in mice. The mice that we're crossing here is a white mouse and a black mouse. Now, even though we've just got two coat colours there, what is actually hidden in this cross are three colours, and that's because of the two genes that are involved, and they are on different chromosomes, and these two give the genetic information that determines the coat colour in this particular cross.
To work at the genetic level, they use mouse blood samples. In genetics, a locus, plural loci, is the specific location of a gene or DNA sequence on a chromosome. A list of loci in a known order, separated by known genetic distances, is called a genetic map.
For each parent, of course, you get two alleles, one from one locus and one from the other locus. So that means you can get 4 by 4 different genotypic combinations or allelic combinations, and that will determine the outcome of the coat colour. And that's most easily explained in a Punnett square, which is a very typical way of writing out the genetic combinations of alleles. If you have two copies of the capital A allele, that's the agouti allele, you will be agouti, the brownie colour. And if you've got two copies of the capital C allele, which is the wild type allele at the albino locus, then you will not be albino. So albino is recessive, so you need two copies of the little c allele to make you white. And then the complication is that the albino locus masks whatever's going on at the agouti locus. So therefore, it whites out everything if the mouse is albino. Now, considering all the possible matings between the male and female mouse, so you have 16 different outcomes. In the top left-hand corner, you've got a double dose of the agouti allele, the white type allele at the agouti locus, and a double dose of the C allele, the wild type allele at the albino locus. So therefore, the mouse is agouti and not albino. In contrast, the albino mouse, as you can see, is actually capital A, which would be agouti if you could see it, but because they're little c, they're albino, and therefore that masks the agouti or black colour, whatever it is. So it's brown underneath, but you never see it because it's masked by the albino allele. So mating the black and the white mouse together produces a brown agouti. And then when you get to the F2, which is what's called an intercross, so here you're mating the brothers and sisters' offspring, the F1s, so all the agouti mice with each other, you can now see the entire phenotypic spectrum, black, white and agouti mice. What's important here is the ratio, which we've worked out from our Punnett square of the outcomes of these coat colours, considering the 16 outcomes, is 9 to 3 to 4 of agouti to black to albino. And that is the first bit of maths down there.
A genetic marker is a variable locus in the DNA sequence with a known location on a chromosome.
So here you can see a map of 156 genetic markers. So at each location we get a genotype for that marker. And the aim of the game is to correlate the genetic outcomes of the genetic markers with the coat colour expressions in terms of mathematics. All the chromosomes are now stacked up one after each other. We've added them together. So we're basically scanning the genome for what's called linkage between the coat colour trait here and the genetic marker loci. On the y-axis is our statistical measure here called the LOD score. A LOD score of about 3 is considered statistically significant, and you can see that we've identified 2 coat colour loci. So we were easily able to map the agouti locus to chromosome 2 where we know it resides, and similarly the albino locus to chromosome 7 where we also know it resides. So maths and stats are incredibly vital in genetics. In my particular area, we're assessing relationships between traits. We're interested in identifying diseases running in families, and the methodology that I've displayed here with regards to identifying the loci for the coat colours, that is exactly the same methods that we use. The mathematical machinery is the same. So we use this technique all the time, and it's a very powerful technique.